the great fortune of hearing Professor Philip Maini, who is, of course, a legendary figure in the area of mathematical modeling of pattern formation. So Philip has an Indian connection. So his parents are from Punjab, who moved to UK sometime in the early 1950s, and Philip grew up in Northern Ireland, and he studied maths and got his BA from Balliol College in Oxford in 1982, followed by a DPhil in 1985, where his supervisor was James Murray, whose book on pattern formation is, of course, a classic in the field. And following postdoctoral research position at Oxford, and after a brief stint as an associate professor at the University of Utah, he returned to Oxford in 1990 and became the director of the Wolfson Center for Mathematical Biology in 1998, the statutory professor in mathematical biology and professorial fellow of St. John's College, Oxford in 2005. So he has been editor-in-chief of the Bulletin of Mathematical Biology and has been a fellow of the Royal Society. And his research includes the modeling of tumors, wound healing, and embryonic pattern formation. And so it's with great pleasure that I invite Professor Maini to give this talk. Thank you, Philip.
Okay, thank you very much. So I'll just try to share screen. So hopefully you can see this, and please do interrupt me if you find that for some reason the Wi-Fi goes out or something. So first of all, thank you for that kind invitation, and I'm very sorry I cannot be there in person. I would have loved to have come, but unfortunately it's the middle of term here, and with teaching commitments and examining commitments, I was not able to come. So I'm going to talk about spatial pattern formation in development. And what I'm going to do is, my understanding is that there's quite a range of people in the audience, from undergraduates, graduates, postdocs, faculty, etc. So this is going to be, to begin with, sort of basic material on this area. But what I will do as we go through is to point out the new areas where there are lots of questions still to be answered. So by spatial pattern formation, I mean this. So for example, here this leopard began as a single cell that then divided multiple times, and then you have all this pattern, like you've got this coat markings here, you've got all the bone structure, etc. So one of the major questions in biology is, how did that arise? So this is it here. How does spatial pattern arise from a fertilized egg, which is almost no spatial pattern? And so not only do we see animal coat markings, we also see, for example, the pattern of our bones. We also see patterns in bacteria, in chemical systems, in lots of different areas. So here's another example. Here's bone pattern in human, humerus, radius ulna, and then the digits. And this type of pattern is quite common in other animals. Here's another pattern. This is a chick, and these are the somites that will form the vertebrae. And these form not all at once, but sequentially. And there's been a lot of work done on how this occurs. So here again are two different, there's the chick, and what you can see in salamander, what you can see here is again 1, 2, and then here you've got 3, here you've got 4. And here's a bacterial pattern. This is a Petri dish, which was filled with some nutrient, which was uniform, and then the bacteria were put in the center, and you can see the bacteria have propagated out to give this pattern. And here's different patterns of same bacteria. And this is a spatiotemporal pattern. This is in a chemical reaction called the Belousov-Zhabotinsky reaction, where two chemicals, where the chemical, depending on the state it's in, it's a different color. And these change out of time, over time. So what would happen is these waves would propagate. These are called target patterns. And here you have spiral patterns, and these would spiral outwards. This is a spatiotemporal pattern. Here's a mother, and here is the child. So the child here is very, very colorful, and as it grows up, it becomes this. So nobody understands how this occurs. This is still an open question. So one of the models, the standard model that's been proposed for this is a reaction diffusion model. And this was, so I want to now briefly describe how you write down reaction diffusion models. So if you have this little volume and there's a surface and you have some chemical, concentration depends on position and time, and then this is the rate of change of that chemical within this volume. So it's equal to flux out, net flux out with a minus sign, plus the net production, integral of net production over the volume. So there should be a V there. So Leibniz theorem, since a volume is fixed, we can take d by dt in, and we get this. And then we can use the divergence theorem as well, assuming that everything is well behaved, to go from there to there. And then we can write this as this. And it is V is arbitrary, then for suitably continuous functions, we get this. And then we have Fick's law, which says that chemicals move down a gradient. Assume that D is constant, we get this. So this is a reaction diffusion equation. And we know that diffusion is a stabilizing process. So if we start off, suppose we're in a one-dimensional domain, but you can extend this to 2, 3 dimensions. And suppose we have a spatially uniform steady state. So in other words, we look for d2c by dx squared equals 0, it doesn't depend on space. And then steady state would be, doesn't depend on time. So we have f of c equals 0, we get that. And let's assume that we have Dirichlet boundary conditions so that this is a steady state. Then if we linearize, do linear stability analysis, we get this equation here. So by substituting the perturbation from the steady state into the reaction diffusion equation, by using Taylor expansion, we get this, and we ignore higher order terms. Using separation of variables and the fact that the boundary condition is C is the constant C star, which means that if we move back here, C hat has to be 0 on the boundary, we end up getting an equation, a solution like this, using Fourier series, separation of variables. So what we can see here is that diffusion is stabilizing. In other words, if I make D large enough, even if this F dash is positive, these will all be negative. And that means C hat will tend to 0, which means that C goes to C star. So stabilizing. And we all know that. Here, if we have water and we pour some ink into it, it will diffuse, and after a while, there's no pattern. So we know that diffusion stabilizes pattern. Then Alan Turing, after the Second World War, he decided to focus on biology because he felt biology was the next big challenge for mathematics. And he wanted to know how patterns formed. So, for example, we saw coat markings, but he was also interested in things like tree branching. So, for example, if you look at the tree and you take a cross section through the trunk, you have symmetry, circular symmetry. And then when you get to the first branch, that symmetry is broken. So he wanted to understand how could that symmetry have been broken. So he proposed that there was a growth hormone. So if you go back here, yeah. So in this case, you got circular symmetry broken. So what he said was there must be chemicals to which cells respond. And these chemicals must, to begin with, be uniform. And then an instability arises in the chemical pattern, and that causes the cells to behave in a heterogeneous way, and you get pattern formation. So in other words, you get a chemical pre-pattern. So in the context of the tree, the idea would be that there's a growth hormone which is expressed uniformly, and then that uniform pattern undergoes an instability. So there's one place where there's now a maximum of growth, and that leads to the branch. And more generally, he called these chemicals morphogens because they give rise to form. So his idea was there would be a chemical pre-pattern or a morphogen pre-pattern to which cells respond. And so his classical paper in 1952 says it is suggested, so the question is, how does this instability arise? And he said, it is suggested that a system of chemical substances called morphogens reacting together and diffusing through a tissue is adequate to account for the main phenomena of morphogenesis. So crucially, what he's saying here is diffusion gives rise to the pattern. Now, we just proved a few minutes ago that diffusion wipes out pattern, and yet he's saying diffusion causes pattern. So in other words, he says diffusion causes pattern. And the key thing is he considered a system of equations. So now if we go through and do our linear stability analysis, and for the sake of simplicity, we'll have zero flux boundary conditions, and then any spatially uniform steady state will then have F and G equals 0. You can take this two-dimensional, or even if it's n-dimensional system, you can write it like this. in vector form, du by dt, where here are the morphogens, vector morphogens. This is the vector of reactions. This is the diffusion matrix, and that's diffusion. And then what he said was that diffusion-driven instability occurs when a steady state, stable in the absence of diffusion, becomes unstable when diffusion is present. So again, let's consider a steady state, especially a uniform steady state. Set W to be the small perturbation. So you could write this as U equals W plus U star. And then the equation for W, dW by dt, following what we did before, Taylor expansion, what we get instead of just F dashed is now we get the Jacobian. So it's all very standard stuff. And so now, when we look for a solution to this of the form e to the lambda t times some spatial component, where the spatial component is an eigenfunction of this. So for example, in one dimension, we'd look for sines and cosines. And then if you had sine kx or cosine kx, the Laplacian would give you a minus k squared. So what you would have then would be dw by dt equals mw minus d times k squared w, where the w here is now the e to the lambda t, the temporal component. And that ends up giving you a problem that is an eigenvalue problem. Okay. So you end up getting, if you look for a solution of the form e to the lambda t, you end up getting this for the lambda, okay, by basically just solving the dispersion relation, where h of x is this, okay? Because basically what you will get, if we go back here, is looking for the solution of the form e to the lambda t, this will then become lambda. So lambda w equals mw minus k squared d times w. You can write all of that in a matrix form, a 2 by 2 matrix times w equals 0. For w to be non-zero, we have to have the determinant of that second order equation to be 0, and that gives you this. And then what we want is, so what we have here is for diffusion-driven instability, we want the steady state to be stable when there's no diffusion. In other words, when there's no spatial effects. That's the equivalent to saying the real part of lambda has to be 0 when k squared is 0, because k multiplies all the diffusion term. And when you plug that in, you get this equation here. It's a quadratic and very simple. Here's the conditions for this quadratic to have real part negative part solutions. Then for instability, we must have at least one k squared non-zero for which this has real part positive. So now again, it's a simple case of we've got a quadratic. Under what conditions does it have a solution that has real part positive? So very, very simple. And here's the conditions we get. These two conditions here. So that gives us 4 conditions. And one thing we find from here is that D1 can't equal D2, because D1 and D2 are positive, and therefore, if they were equal to each other in condition 3, we could divide through by the D, and then we find that is inconsistent with condition 1. So that gives us a condition that for diffusion-driven instability to occur, the diffusion terms cannot be the same. It also tells us that these Jacobian matrices must have a certain sign structure. So for example, they can't both be of the same sign, because if they were both of the same sign, they'd have to be both negative to satisfy this, and they'd have to be both positive to satisfy this. So one has to be positive and one has to be negative. And then if, for instance, this is positive and this is negative, then this is negative, this product is negative, which means this product has to be negative to satisfy this condition here. Because if this product was positive, then we'd have negative, negative, that's not going to be greater than 0. So these must have different signs. So what this does is it gives the restriction on the types of kinetics that you can have. That they have to have certain types of interactions. And so if we look at A here, this is dF by du, and dF by du being positive means that U activates itself. If it increases itself. And here, dg by dv being, dg by du being positive means that it increases v. Because you think about it, we would have dv by dt equals gu times u. And if gu is positive, it means that u is increasing v. V is inhibiting u, because this is df by dv times v. So that's negative, means the more v there is, the lower u is. So u is activating v, v is inhibiting u. Moreover, if we go back here, if df by du is positive, dg by dv is negative, the modulus of df by du must be bigger than the modulus of if df by du is positive and the gv is negative, then the modulus of this must be larger than the modulus of this to give negative. And that means here d2 must be larger than d1, which means that v must diffuse more rapidly than u. So then we've got the activator, which is u. So I've got these the wrong way around. du should be big, d2 has to be bigger than d1. So the activator is, which is u, doesn't diffuse as much as the inhibitor, which is v. And so we have short range activation, long range inhibition. With zero flux boundary conditions, Fourier series expansion must have cosines, and they must be n pi x over L. These are admissible modes, And so k squared is n squared pi squared over L squared. And so what we do is we have our dispersion relation has to look like this, and the admissible modes lie between these two values. So what we can see here, these two values are fixed by the parameters. And so if we increase L, we have to increase N, in other words, to lie within this domain to satisfy this condition. So as L gets larger, N gets larger, which means we get more complicated patterns. And as if L got very small, then we might not be able to satisfy this, because even for n equals 1, if L is very small, we can't satisfy this condition, which means we have no admissible mode. And that means even though the dispersion relation looks like this, the first admissible mode is outside the range that can go positive, and therefore we do not have the instability. If we go to two spatial dimensions, the Fourier series, the admissible mode is this, and now we get the k squared must look like this, where A is the domain length and B is the domain breadth, you can think. So this is in the x direction, this is in the y direction. And so what we can see here is if B gets very small, so we go from a sort of almost like a stripe, then M has to be 0. Because if M is 1 and B is very small, this would be huge and therefore wouldn't lie within the k squared values for which lambda is positive. So if M is small, so M has to be 0. If M is 0, this is 1, and we're back to the one-dimensional case. So in other words, what that means is that if you have a very narrow domain, you can only have pattern changing in the x direction. In other words, you can only find stripes. If the domain, however, B is large, so that you've got like a square domain or something like that, then M can also be non-zero, and then you've got pattern in two x and y, and those are called spots. So this is what is called a developmental constraint. So what this tells us is that you need a minimum domain size for pattern. Pattern complexity increases with domain size, and you have something called development constraints. So some examples of F and G, these are not particularly important. This is a Gerr-Meinhardt model, where for U and V and everything else is a positive constant. He just developed that so that the partial derivatives would have the correct sign structure to give the pattern that you wanted. And what you can see here, U is causing V to increase, but V is inhibiting U. This is the Thomas model, an empirical model where he fit these functions to experimental data on oxygen and uric acid. And then this is the Schnackenberg model, which is a special example of another Meinhardt model in which he wrote down some chemical reactions and got these types of terms. But there's lots of different models. And linear analysis turns out to be a pretty good predictor of patterns in many cases. But there are many cases in which it's not a good predictor. And this is still an open question. Why is that a case? So Turing patterns have been found in chemistry. So De Kepper and Swinney have found, this is called chloride iodide malonic acid, and they found Turing patterns, and Langell and Epstein have modeled it with the reaction diffusion Turing model. This is the reaction scheme here, and these are the patterns, the experimental pattern that they found. Now let's go back to the predictions, and Jim Murray's book, which is mentioned at the beginning, is a wonderful collection of this material. And so Jim here has simulated the reaction diffusion system on this type of structure. And here the domain is getting larger, but it's being rescaled because otherwise it wouldn't fit on the domain on the page. So here you see there's no pattern. And here you see this would be the equivalent of a cosine. So the idea here is where the morphogen is above a threshold, label it one color. Where it's below a threshold, label it another color. Domain gets larger, more complicated pattern. And now you can see how the linear analysis no longer holds, because as the domain gets larger and larger, the pattern does not continue to become more complex. And the reason for this is that in this case, there are multiple modes that are unstable and they're interacting with each other, and we've got non-linear interactions occurring. There may also be subcritical bifurcations occurring here. We don't know. Here's an example of that one, the Valley Goat. Here's an example, the Belted Galloway. And here's an example here that you see as the domain narrows, the pattern becomes simpler, and these are consistent with looking at eigenfunctions of the Laplacian on a tapering cylinder. So in this paper here, Shubin and Albert, who were experimentalists, looked at a lot of animal coat markings, and what they found was a developmental constraint that in most cases, if the complexity on the tail could not be greater than the complexity on the body. So you could have spots here and stripes, but you couldn't have stripes here and spots. So that's what the data were telling them. Meanwhile, Oster and Murray, using mathematical analysis, using the Turing model, showed that if the parameter values are such that here the only eigenfunction that became unstable was one that had stripes, then on the two-dimensional domain, you could not get more complex patterns. And now in football shirts, the same thing happens. Here you look, stripes and no pattern. Here, same thing. This is the male football teams in the UK. This is a bit old now. You see in all of these patterns, the pattern on the jersey, on the body, is not more complex than the pattern on the arms. And this is rugby, the same thing. So I do wonder whether this developmental constraint that surrounds us in nature sort of in our mind gives us an idea of what appears to be normal, what appears to be beautiful. And unknowingly, we design our clothes based on what we see in nature. But that's just a bit of a side issue. Okay. So here's an experiment. Remember, if you have small, if you were to decrease the domain, would you have smaller digits? For instance, if you were to look at digit patterns, or would you have fewer digits of the same size? Turing predicts you should have fewer digits of the same size. So that's what happened. This is salamander and this is frog. What they did, this is normal. And what they did early in development, they reduced the ability of the cells in the limb to proliferate. So you got a smaller domain and you get fewer digits, like the Turing model would predict. If you do the opposite, this is the chick. Normally you'd have 3 digits. Here an experiment was done to make the limb wider. Now the question is, will you still have 3 digits, but they'll be thicker? Or will you get more digits all the same size? Turing predicts the latter, and there you go. Here's just another very nice example. If, from Murray's book, if patterns are really eigenfunctions of the Laplacian, then what they did was they took metal sheets that were shaped like an animal coat, and they vibrated it, because we know that vibrations of a drum or a string are eigenfunctions of the Laplacian. The amplitude satisfies eigenfunctions of the Laplacian, the spatial part of it. And what they did was do holograms of that. And what you see here are the eigenfunctions, and they look like animal coat markings. Now, there are counterexamples to Turing. Look here, stripes, followed by spots. Here's no pattern, followed by stripes. So what could be happening here are nonlinear effects, subcritical bifurcations, or different parameter values in different parts of the domain. And these are problems that haven't been addressed yet. It's still an open question. Likewise, this is an open question. As the domain got larger, the pattern should stay the same, or it shouldn't get simpler. And here it's got simpler. There's no explanation for this. It's an open question. One thing I should say is that the cells only have a certain time window in which to respond to these chemicals. And then after that, their fate is determined, and all they do is they divide. They don't change their fate again. This is Hans Meinhardt, did this beautiful book called The Algorithmic Beauty of Seashells. And what he shows is having seashells growing and having reaction diffusion equations, a little bit more complicated than what we've looked at, on these growing domains, more complex curved domains. You can get patterns. This is the real, and this is the model, real, model, real, model. You can really capture a lot of these patterns. So pattern can arise as an emergent property. 2 stabilizing processes can give rise to an instability. Turing proposed the idea of morphogens, morphogens have now been found. Turing patterns arise by diffusion-driven instability in chemistry. The question is, do they exist in biology? Now, these morphogens, in many cases, they have the activator-inhibitor interaction that's necessary for a Turing pattern, but the ultimate unrefutable proof that the patterns in biology arise due to Turing instabilities is still an open question. So we've got morphogens, we've got activator and inhibitor, we've got those in biology, we've validated Turing's model in chemistry because it's easier to control things in chemistry. The unrefutable validation in biology is still a challenge that lots of people are working on. So another model for pattern formation is Wolpert's French flag model. So the idea that Louis Wolpert had was there was a source of a chemical or morphogen, and it diffused across the domain. Where it was high, these cells would differentiate into one cell type, in this case red. Where it was medium, these cells would differentiate, become this color, here become this color. This is called positional information, and indeed, he proposed this for limb development. So these would be the different digits that you see in the chick. And in fact, such a chemical morphogen was found called retinoic acid. Now, Wolpert, he wanted to disprove Turing's model. So what he and Anita Hornbrook did was they took a slice of the limb where it gives rise to the humerus, and they took this part, the anterior part of a donor, or anterior part of a donor, and they replaced the posterior part of the host by this part. So the domain remained the same size. So the Turing model would predict that the pattern would remain unchanged. And the pattern is one humerus, but they got two humeri. So they proposed this to invalidate Turing's model. So they constructed a new limb bud which had the same size as the normal limb bud, but it gave two bones instead of one. The Turing model predicts that the domain size does not change, the pattern should not change. So what we did, we combined Wolpert's model with Turing's model, and we said, suppose there was a gradient. And in this case, we said, suppose it was a gradient of a chemical that affects the diffusion of one of the morphogens. So the diffusion looks like this. Then what would happen if we did the experiment that Wolpert did? This would be normal, and this would be the result of the experiment. We would get double anterior limbs. So we said, if there was a gradient of diffusion across the limb, this would give rise to, couple that with Turing, you would get the result that Walpert and Hornbrook got. While we were doing this work, a paper came out that said retinoic acid, an experimental paper saying retinoic acid modulates gap junctional permeability. So remember, what I said was that Walpert had proposed a morphogen gradient, and this retinoic acid produces that gradient. Crucially, that, if we go back here, the retinoic acid is produced here, and it has a gradient like this. And what the biologist showed was that retinoic acid inhibits diffusion. So if there's a gradient of retinoic acid like this, that corresponds to a gradient of diffusion like this. Exactly what our model predicted. So it's consistent with our model. But what this shows you is that it's much more complicated than simply having two chemicals reacting and diffusing. There are sources and sinks of various chemicals on the boundary that are affecting the parameters of the Turing model. And there's a lot of work going on combining the positional information model with Turing's model and with experiments. There's a lot of work going on at the moment in the limb to try and see how do these couple to give rise to the patterns that we observe. Now, there's also, of course, domain growth. And this is one thing that makes biology, one of the many things that makes biology more complex than engineering. In engineering, things don't grow. In physics, things don't grow. This is classical paper by Kondo and Asai, where they were looking at this pomacanthus, and as the pomacanthus grows and these stripes get further apart, a stripe appears in the middle to preserve the wavelength. And this is exactly what you would get from a reaction diffusion model. As the domain grows, you get insertion of new patterns. And this appeared on the cover of Nature in 1995. And you could get this other type of bifurcation, where a zipper bifurcation, where you get the stripe forming like this, where this propagates along and you get this type of pattern like this. And these are the type of patterns you see if you take pictures of this animal over time. Now, one thing that's known about Turing patterns, and that's been known for a very long time, is that they're not robust. Small changes in initial conditions can lead to changes in pattern, and therefore that was one of the major arguments against the Turing model. Here's an example of that. What we did here, this was work with Edmund Crampon and my colleague Eamon Gaffney, was we took a domain of a certain size. Let's go down to this one here. We chose the value of the domain so that we would get mode number 15. So this is in one dimension, so we want cosine 15 pi x to be the pattern. And then we run it, see what pattern we get, and then put different initial conditions, see what pattern we get, continue, continue. And what we find here is, yep, sometimes we get 15, but in fact, more often we get 14, higher frequency, 13, 12, no 11, we can get 16 and 17. So that's an example, an illustration of the pattern depends on the initial conditions. This is not good. But what happens if we let the domain grow? So we let the domain grow, we go through the same analysis, and we get this extra term in. And now we find, if we have the Gere-Meinhardt model, we have the domain growing exponentially. Now you see what happens. We have 1, and then we get sort of 2 patterns here, and then we get an insertion, and then we get 2 insertions, and then we get 4 insertions. We get a period doubling. And you can see here with the different process, we get a pattern doubling. So this means that if we wanted to get mode 16, for example, and we started off with the domain mode 16 is this one. Here, we started off with a domain that would give us mode 16. A lot of times we would get mode 16, but sometimes we'd get 15, 14, or 17. But if we let our domain grow, we will always get 16. So this is very robust. Now, it doesn't mean that the domain has to physically grow. What it means is the domain in which pattern forms has to grow. And we see a lot of cases in biology where pattern forms behind a propagating front. And there's work we're doing at the minute, which is in review, where we try to understand how does that pattern depend on the speed at which this front moves. But what would happen if you wanted to get mode 15? You can't go double and get to 15. But you could get 15 if you had growth just at the top. Then you'd have 1, 2, 3, 4, 5, 6. Keep on going to get 15. So what this shows you is that growth can enhance robustness at the expense of others. And it doesn't have to be the growth of the domain by physical growth. It can be a chemical that grows. And in fact, what some people have done, there are some chemicals whose reaction depends on light. So what they can do is they can have the reaction going, and then they can change the light and have the light switch on as a traveling wave. And then you see the pattern form. And there's a question then of, as you can then change the domain geometry in which the pattern is forming. Make it circular, make it square, make it rectangular. And what the light is doing is it's switching the parameters and moving them in to the domain in which pattern forms. So this is a lovely way in which we might be able to validate our models. And there's a lot of work going on that. And what I would say is that a lot of times when you talk to people about Turing's theory, a lot of people say, oh, that's all been solved. This is a collection we did 3 years ago. Andrew's the first author on it, in which we have a collection. We invited lots of people to submit papers on what are the key unanswered questions still on Turing pattern formation. And what we have here are things like, how does the domain geometry affect the growth of the pattern? How does different chemicals, spatio-temporal variation in parameter values, which are known to happen in biology, how does that affect the patterning? Are the patterns all due to subcritical bifurcations or supercritical, or could there be subcritical bifurcations? For example, if you have different boundary conditions, does that influence the patterns that you get? And how do the patterns interact with each other in the nonlinear regime? And there are many cases where you've got a patterning process happening in one layer of tissue, a patterning process happening in another layer of tissue. How do they interact? You also have non-local interactions, and these lead to integral partial differential equations. And there's a lot of work going on there. How do we analyze these? And in fact, what I've done was I've gone through here. I've said, well, you can do all this in one dimension, but it easily generalizes to more than one dimension. But when you have, and that's because the eigen functions generalize. When you get into non-local effects, there are certain situations where that doesn't occur. There's not that generalization of the Fourier transform. And what you can get then is that patterns or systems that are stable in one dimension, but they're unstable in 2 dimensions. And that's work we've been doing recently with the graduate student June Jowell and with Andrew Krauser and Eamon Gaffney. And this leads to a whole other set of problems that need to be addressed. So for the last few minutes, last maybe 20 minutes, what I will do is talk about where patterns form due to traveling waves. And I'm going to focus on something called the neural crest. So the neural crest, this is a transient structure in vertebrates that give rise to a lot of very important organs in our body. So for example, in our gut, the lining of the gut is formed by neural crest. So these cells leave the neural tube, which is a precursor to our backbone, and they move along the gut, and they have to cover the gut to give it the right mechanical properties so that it can, the gut can function properly. If that doesn't happen, you get diseases like Hirschsprung's disease. There are 4 neural crests, and this is just a cartoon. Here's head, here's tail, here's the neural tube, the sort of backbone, and these are moving in corridors. And here is it happening. You know, if you look at a cross section, here's a neural tube, and the domain is growing. You've got a curved domain, and these cells have to move and reach these areas here. So the reason for studying this is that 1/3 of birth defects arise when this doesn't work properly. Moreover, some of the neural crests are very similar to highly aggressive cancers. So if we could understand how does this movement, how is it controlled, then maybe we might be able to control and stop the movement of cancers that derive from neural crest cells. Moreover, the neural crest is experimentally tractable. You can do lots of experiments in a way that is very, very difficult to do with tumors. And here's a paper in which we just summarize some of the mathematical challenges of this system. So this is work, a long time collaboration over a decade with Paul Kulisa and Rebecca McLennan. They used to be at the Stars Institute for Medical Research in Kansas. They're now at the Children's Mercy Hospital carrying out research, this similar research. And with Ruth Baker, my colleague here, and David Kay also here, graduate students, Louise Dyson, Linus Schumacher, and Raza Gennati, who've now moved on, Duncan Martinson, who's still here in Oxford, and some of the latter work with my colleague Helen Byrne. The main collaborator in all of this being Paul, Ruth, and graduate students. So let's go back here. What we do, we're looking at what's called the cranial neural crest. So here's head, here's tail. These come out of rhombomer 4 and they go to branchial arch 2 and they form a chain. And this will eventually give rise to a lower jaw. So cranial neural crest means giving rise to parts of the head. So what we did was we said, okay, it's three-dimensional, but this normal to the surface dimension is very, very narrow, so we'll assume it's 2 dimensions. We will assume that the curvature is small and can be ignored, so what we have is a fat rectangle that's growing. So the rectangle is growing in this direction here, and these cells are coming out and they move along. And how can they form a chain? And here are just measurements of the growth of the domain. Here's the length of the domain, and over a 24-hour period, the domain grows about 1 millimeter in length. And these cells, which are in their hundreds, roughly speaking, move along that domain in a chain. So it's known that if, it's sort of obvious, that if these cells were moving randomly due to diffusion and advection due to the movement of the domain, they wouldn't form a chain. They would move randomly to the end of the domain, but they wouldn't move in a chain. So what our biology colleagues noticed was that this chemical called VEGF was produced by the tissue. And it's known that the neural crest cells will consume VEGF. That creates a gradient in VEGF, and these cells move up the gradient of VEGF. It's called chemoattractant. So that leads to a biased random wall. So their proposal was, could this give rise to this chain behavior? So what we did was we wrote down an equation for VEGF So here it is here. C is VEGF. The domain, the length is, we non-dimensionalize this, so the length is just changing in the x direction. So this is the length. And this very complicated looking equation is simply the equation that fits that growth curve. That's all it is. We have a production term that, if nothing else was going on, would give you VEGF at level 1 as a steady state, non-dimensionalized. Because we have domain growth, we have this extra term, but that's derivative and that's dilution. We model the cells as disks that are centered at xi, yi, and they consume VEGF. With here, we just take this to be something that's maximum in the center of the disk and decreases as you move away from the center of the domain. And then we put either Dirichlet boundary conditions or zero flux boundary conditions. And what we do then is we assume that the cells are discrete entities. Stochastically, they reach out and sense the domain VEGF near them. If they sample VEGF concentration larger than at their center, they then move in that direction with the speed given to us by our biology collaborators. And what we find is the model doesn't work. So here's the domain at the beginning. Here's cells coming in, sensing the direction. They come in when there's space. So this is a volume exclusion process. They start moving, and notice the gradient is very localized. So afterwards, the VEGF is all consumed by the cells, and now they're just randomly diffusing and being advected, and therefore they don't form the chain. We get an idea of this from classical Keller-Segal model for chemotaxis, where N is cells. This is moving up the chemical gradient. This is diffusion. This is production of the chemoattractant. And this is sensitivity to movement up the gradient. This will give you a pulse, traveling pulse, just like we got. For it to give a coherent chain, this term has to be singular of this form. And this is not realistic biologically, to have the sensitivity go to infinity as the chemoattractant goes to 0. So what our student Louise Dyson did was she asked, suppose there were 2 cell types, leaders and followers, and the leaders respond to the chemoattractant, move up the domain, and the followers, here in white, stick to the cell in front of them. So they'll form these chains. So what the biologists did was they then went back and they analyzed a lot of genetic analysis of these cells. And what they found was the cells at the front upregulated genes that were to do with sensing the chemoattractant, moving into new areas of the domain, breaking down the material so they could invade, while the cells at the back upregulated cadherins, which are to do with stickiness, adhesivity. The cells at the back are stickier than the cells at the front. in agreement with the model. But in fact, it's much more complicated than that. It's not two cell types. If you go through and look at the gene expressions, they vary all along the domain. So one thing we're doing at the minute is analyzing this to see, for example, if you've got a gradient in the domain, in the adhesivity, does that enhance the movement and the coherence? Now, there are many, many more complications. The growth is not spatially uniform, and it is stimulated by the cells as they move through the domain. There's the gradient of phenotypes. We imposed Dirichlet boundary conditions, and then we also considered zero flux boundary conditions. When we did this, there were no biological justification for doing that. Recently, our model predicted that if you had boundary conditions, they didn't have to be along the whole domain to confine the cells. They had to only be along about half the domain. And so our colleagues started to look in the neighborhood of the domain near the neural tube, and they found the expression of various chemicals that inhibit movement of cells. So we're in the process of writing a paper where we are modeling those effects. In our model, leaders can break away. So our model was suggesting, could there be something that slows the leaders down so they don't break away? Our colleagues went and they looked at the domain near where the cells come out, and they found the expression of a protein that slowed the leaders down, but then vanished so the followers could catch up. They also then applied that protein to melanoma cells, and they slowed the melanoma cells down. So that's an example where, by learning from developmental biology, we could translate our findings perhaps into disease. Okay. And here's a number of papers that we've written. This was the first model published in 2012. Here is the idea of having the leaders and followers, which is 2015. Here is more of a review article. Here's the article where we discovered the expression of this DAN protein that slows down, restrains cell movement. It's 2017. Here's a model. Here's a paper in which we say we present the challenges, the modeling challenges. This paper here is where we find out that domain growth is not uniform. And here's the recent paper in which we find out that these chemicals are expressed on either side of the corridor that's confining the cells. And although we discussed stickiness as being one example in which the followers move in response to the leaders, there's another idea, which is that the leaders might create a tunnel through which the followers move. And our collaborators found that the leaders upregulate something called aquaporin 1 which enables them to create a whole tunnel. Aquaporin 1 is also upregulated in tumor cells. So hopefully what I've done there is given you an idea of some problems that are in mathematical biology, giving you an idea of two different methods of addressing them using PDEs, using cell-based models, and trying to indicate that while a lot of progress has been made in this area, there's still a huge number of open questions that need to be answered. And so I'll stop there and thank you for your attention.
Thanks, Philip, for a wonderful talk. Uh, Time for questions.
Do you have any insights on how this pattern formation could be used in the case of camouflage in chameleon or octopus?
Yeah, so that's a very good question. So the thing in those cases, what happens is the cells themselves change color. So it's not like the one we've been doing here, where the cell becomes, something that produces black and therefore continues to produce black. In those cases, the cells themselves can change color. And I know there's quite a lot of biology that's gone into that. I don't know if anybody's modeled that because it's not something I've looked at. So there's something going on inside the cell. And the question is, I don't know whether that's also accompanied by the cells moving and reassorting. That's my ignorance of the area. So I'm sorry that I don't know the answer to that, but I do know there has been quite a lot of biological work on that. And there may have been modeling, but that's not something I know about.
Shailaja?
I was wondering about the neural crest migration model. So neural crest migration occurs over several hours, and along the axis, different elements form of the jaw, the ventral element, the joint, and then the dorsal element. So does your model propose that there are different populations of leader cells for each of these different subsets of neural crests along the DV axis?
Yeah, so we just looked at the cranial neural crest cells. So we haven't looked at the other cells. What we do know is that there's very little crossover between the cells moving along rhombomer 4 and the other cells. Right. So we don't know whether there is mixing and whether what's happening in the other cells is the similar sort of thing that's happening, because we've just looked at the cranial neural crest from rhombomer 4 to branchial arch 2. So we don't know whether it's the same leader-follower behavior happening in different places, whether the same genes are being upregulated, downregulated, whether there's the same cue that's going on. That's something that, I don't know. It may be something that other people have looked at.
Yeah, I was wondering.
Yeah.
No,
sorry, go ahead.
I was wondering, in the branchial arch 2, the stream that enters branchial arch 2, is a leader cell population fixed, or like in other systems, it's known that for some time a population is a leader cell and then they fall back and a new population of leader cells take over. So is that known to happen in the crest, neural crest, cranial crest?
So, yeah, actually, thanks for pointing that out, because one thing I forgot to mention here, which is a very important thing, is that there is phenotypic switching occurring in that leader cells can become follower cells. And so, for example, if you put VEGF, a source of VEGF, along the corridor, the stream will split up, and some of the follower cells will respond to the gradient and move up.
It'll take over
as gradient. Yeah. And so what we're hopefully doing now is Paul wants to redo that experiment and then do a genetic analysis to see have those cells really become leaders.
Okay. Okay.
So that's a big, that's an important issue, because then that would also be important in terms of the robustness
Correct.
of this process.
Correct.
That if for some reason a leader gets left behind, can a follower take over and become? And in fact, why we noticed this in the first place was one of the experimental procedures we did to test the mathematical model was to take follower cells from a donor and put them ahead of the leader cells in a host. And now our model predicted that those are followers, there's nothing for them to follow, so they just randomly move.
Yeah.
And what the experiment showed was that they actively start moving. They create a gradient and move. So what Paul's going to do again is to now analyze those cells by redoing those experiments to see, have they upregulated the leader genes, because they seem to be behaving like leaders.
Yeah. Okay.
So we're hoping to really do a lot more along that.
Okay. Thank you.
So I have a question related to the part where you discuss pattern formation in a growing domain in two different models, namely the Meinhardt and the Schnackenberg model.
Yeah.
So in Meinhardt, you see stripe insertion between two
Yeah.
stripes, but on the other model, you see splitting of stripes. So I was just wondering, what's the basic difference in the mechanism of pattern formation between these two models?
So what happens in one of those models? So depending on the structure of the Jacobian, whether it's plus plus minus minus or plus plus minus minus, what will happen is the morphogens will either form in phase or they will form out of phase. So in other words, either U and V will have maxima and minima that coincide, or the maximum of one will coincide with the minimum of the other. So in the activator inhibitor model that I talked about, where there is a lot more activator, that will produce a lot more inhibitor, but then that inhibitor will diffuse more and inhibit the activator. And so in that way, you can see that the U and V, depending on how those interactions occur, can either be in phase or out of phase. And what we think is happening is that these different, like the branching or the insertion, is arising due to those different in phase or out of phase. But how to actually prove that is something we're actually trying to do at the moment in a different context of not growing domain, well, not physically growing domain, but a domain where the domain over which pattern can form is increasing due to the movement of another chemical. And we're trying to figure out, can we understand how you get branching versus insertion?
I have one more question, if you don't mind.
Yeah.
So the robustness of this pattern formation in the growing domain, do you also expect to see this robustness in a 2D or higher dimension and also on curved manifolds?
Well, in 2D, it's been shown. So for example, in the chick, the positions where the feathers form in the back of a chick, they're roughly speaking hexagonal. But that pattern doesn't arise all at once. What you get happening is, first of all, you get the spots where the roots, where the feathers are going to form, first of all, appear in a line of spots along the backbone. And then they appear on either side, but out of phase spots. And then the next row appears, but out of phase. And in that way, you get hexagon. And what and there's been a number of models that, I mean, if you were to just take that whole domain and simulate a Turing model, sometimes you'd get hexagons, but most of the time you would get a mess. You would get sort of defects, just like in convection flows that you get a mess. But if you have a domain where you shed light, if you take the model and you have the propagation happening, then you will get these patterns forming in a sequential way and get the robustness happening. But then the question is, what is that wave that's moving along? What is that? It's essentially meaning that the parameters behind the wave form Turing pattern. The parameters in the model ahead of the wave are outside the Turing regime. And then as you move, those parameters move into the Turing regime, and therefore pattern forms. But the question is, how is that happening? And people have proposed mechanisms for that, but that's still an open question. So the theory, I mean, numerically, you will get the robustness. But again, it depends on how fast this is moving. And that's something we're looking at, because how fast relevant to what? We don't know. What we're wanting to do, we're actually doing this, like using envelope equations, stuff like this, trying to figure that out at the moment, as when you get insertion versus splitting, even in the one-dimensional case, it's still an open question.
Continuing on the previous question that Soling asked, do you also have situations where you see arrest of patterning, like something analogous to clock and wavefront model? So instead of a growing domain, you have a wavefront that freezes the pattern before it and increases the domain size for more pattern formation to occur.
Well, this one here, I mean, the clock and wavefront, which, as you know, has been proposed for the somites, there hasn't been one proposed for this one. In fact, when I did my doctorate, we had a different model. There was a Murray, Oster, and Harris model, where the idea was that due to mechanical interactions, you got aggregations of cells. And we had as our bifurcation parameter the force exerted by cells, attraction force exerted by cells. And what people had observed was that the traction forces increased with maturity. And that was our model, that initially the cells along the backbone are formed first, maybe, and therefore they're mature. And then there's a wave of maturity that sweeps across the back as the animal is growing. And as that wave of maturity waves across, then you get you go through the bifurcation, and therefore you get the patterns to form. But whether that is the case, it's still an open question. As far as I know, nobody's looked at that to see what that is. Yeah.
But you would need something more than Turing to arrest the pattern, right? Even if you change the parameters once the wavefront has passed through, like, inhibition of diffusion, I think the pattern would right?
Well, the thing is, this pattern would form and then it would stop when the wave stopped. So I think I'm misunderstanding your question.
No. I mean,
the pattern would form behind the wave, and then it would stop when the wave stopped.
Yeah, but that's not arrest. You're not arresting the pattern, right? The parameters behind the wave remains constant, right, throughout the domain.
That's right. Yeah.
I'm talking about something like a moving pulse. So can you arrest?
No. So Yeah, so we haven't really, that's not been proposed yet. So it's been proposed that there is, it's just a wave. And so behind the wave, the model, the parameters are in the Turing regime. So you get the Turing pattern. Ahead of the wave, there is no Turing pattern. And then as the wave moves along, then, the Turing pattern sort of fills in. But the question is, going back to the last question, in one dimension, you can show that then that'll give you peaks and peaks and peaks. In two dimensions, there's a question that if you've got a nice pattern here and the domain moves along, does that pattern get destroyed before this next pattern can come up? And that's something that people haven't looked at. That's still a question. And that's where what we're looking at is a playoff between the time it takes for the pattern to form in the Turing system and the time it takes for this wave to propagate. Which leads to the further question of what is the time for a pattern to form? What is the definition of that? It's never been, nobody knows. And that's precisely what we're trying to do, even in one dimension. It's proven to be extremely complicated by trading off the timing of this pattern being set and the timing of the movement. And it's a very good question. Nobody really knows. I mean, biologically, nobody knows, and mathematically, nobody knows. Yeah.
Just one more question. You mentioned that to induce instability, you need the condition where at least one of the eigenvalue is positive once you add diffusion.
Yeah.
Does it say something about the system if we know how many eigenvalues are positive, or that's not relevant to the?
No, so I mean, the only thing that happens is that probably there will be, I mean, this idea of isolating a mode is a very, very special case. And depending on the parameter values, there might be lots of modes that are growing, and then they would interact with each other, and then you'd get some complex pattern. So in fact, one thing one could try to do, so the question is that when you look at a pattern, I mean, if it's like a clear stripe pattern with a clear wavelength, then you can say, okay, it's isolated this mode. But if you look in general at a pattern, like a pattern on a zebra, it's not, very strict stripes. It's sort of angular and all this and this sort of thing. If you look at the spots on the leopard, it's not hexagonal. It's sort of all over the place. So there's a question then of how do you characterize that pattern in order, like, do you take a Fourier transform of that to see which modes have been upregulated, or do you take a model and you do some topological data analysis and then do model fitting to get the parameter values, and then see with these parameter values what is the dispersion relation. And now that is also an area precisely that people are looking at. How can you fit the model to the data, which is a spatial pattern? It's a completely new area of research using topological data analysis and everything that's ongoing.
I'm slightly unrelated, but one more question. Do you also look at coupled systems? Like you have two different Turing systems that are coupled. And do you know if this has any implications or relevance in biological systems? So to get more.
Well, we have looked at those. Yeah, we have looked at those in the context of, again, having the epithelium and the dermis, so the skin, and having one sort of reaction diffusion system in that, another reaction diffusion system here, and then having the coupling there. And one way, an easy way, the pattern that can give is a mixed mode solution, because there are some cases where patterns look like there are two patterns of different wavelengths that have been superimposed on each other. And then that's a type of model that, at least in the linear regime, would give you that pattern. And it is known that there's certainly, like with germs and stuff like that, there is some crosstalk between the layers, the epithelial layer and the dermal layer. And how is that affecting, is it helping to stabilize a certain pattern? Is it helping to stimulate a certain pattern? There's definitely crosstalk going on. Yeah.
Okay, thanks.
Yeah.
All right. So, Phillips, thank you so much for, spending time with us. And I really look forward to your visit to India next year, and hopefully you'll be able to touch Chennai on your way.
Yeah.
And let's thank Philip one more time.
Thank you. And thank you very much. All those questions really show how much we still need to do and what an exciting area this is to work in, because questions touched on really important areas we need to look at. Thank you. And thank you for your invitation.
Bye.